I'm Chloe, and I designed RNA vaccines targeting Alzheimer's disease, or AD, using single-cell RNA sequencing data and proteomic analysis. AD is a neurodegenerative disease that is becoming more and more prevalent due to aging population, yet there's currently no cure, which led us to the idea of vaccine. We reason that, like COVID-19, RNA vaccine is optimal because it does not integrate into your genome like DNA vaccine, and it can encode longer peptides compared to subunit vaccine. So our strategy is twofold: conventional targets and novel targets. For conventional targets, we put our focus on A-beta and Tau, which are two key protein aggregate hallmarks of AD patients. We used protein blasts to identify any possible off-target effects, and then we devised a scoring algorithm based on a disease-specific post-translational modification, or PTM, dataset. For novel target, we located candidate genes with a computational screening of single-cell RNA sequencing data from Allen-Bragg-Alice and also TRAPSeq database that's recently published on Cell. We identified genes differentially expressed in AD, their hub genes, and also the gene that's uniquely found in regions most easily affected by AD, or the most susceptible regions. After we located candidate genes, we output candidate vaccine sequences using tools such as Protein Blast, AlphaFold, which is a deep learning algorithm that's recently published last year on Nature, and we also used more single-cell RNA-seq data to do that. So moving on to results, for A-beta, we identified using BLAST that it is a key part of a important synaptic protein named APP. Hence, the apparent cleavage of A-beta will likely impair normal brain functions, which would be counterintuitive to our purpose. And second, our Tau vaccine algorithm specifically gives every amino acid a position score based on its AD-specific protein modification, its AD-specific clustering, and also the differentially expressed genes in AD patients. Our algorithm revealed that the shorter the sequence, the more AD-specific or the more effective the vaccine will be. However, if a vaccine is too short, we would risk mistargeting other proteins. Hence, our ideal sequence is 7 amino acid long, which is KPV-DLSK as outputted here, and is also visualized above using AlphaFold. And on the other hand, for novel targets, our computational screening reveals 3 top candidate genes listed on this table here, yet 2 of them have been reported to demonstrate significant physical defects in their knockout mice models. Therefore, they are not ideal targets. However, S100A11 is a gene that does not exhibit any obvious phenotype abnormality in its knockout mice models, hence it became our ideal target gene. Therefore, we conducted further analysis with single-cell RNA sequencing data, and we found that not only is it present in susceptible neurons, it is also primarily found in a subtype of microglia and also in monocytes, which would aid us greatly when we try to design the vaccine engineering part. And we then output candidate vaccine sequences of S100A11 by identifying 5 non-overlapping regions of this protein and other S100 protein family, and the vaccine sequences are again outputted here by AlphaFold. In conclusion, although I didn't find an A-beta vaccine, I did identify a Tau vaccine sequence, and I also identified a novel target, S100A11, for the first time, and offered several candidate vaccine sequences for further testing. My algorithm may also be widely used to find vaccines for any neurodegenerative disease with disease-specific protein modification data. Another interesting observation is that the beginning and the end of the Tau vaccine sequences are AD-specific ubiquitination sites, giving rise to a question of why are these Tau amino acids not degraded. It would be an interesting question for further research. And meanwhile, I'm combining this research with spatial transcriptomics data recently published on Cell that directly visualizes the plaque genes in AD patients. This data about brain connectivity will allow me to better understand my vaccine's effects. Lastly, the proposed vaccine of Tau and S100A11 may also be utilized in wet lab to test in mice for their efficacy. And meanwhile, S100A11 would be a promising target for AD therapeutics development. And that's all the references I used for this study, and thank you so much for the attention.